Welcome back. In this last part of the lecture recording, we are going to be looking at DNA fingerprinting. We are going to look at something called gel electrophoresis and the application of that. And we will finish the lecture by talking about gene therapy. So let's start with DNA fingerprinting. So there's this concept of polymorphism. Polymorphism is any differences you can see in a DNA sequence between two individuals. These differences can occur in the coding regions of your genome, so that exists in a gene level. When you have polymorphism in a gene, that creates different versions of the gene, the alleles in the genes. So someone could have brown eyes or blue eyes. That's considered as a polymorphism. But the polymorphism can also occur in non-coding regions. So these are regions in the DNA that is not part of a gene. In the non-coding regions, variations may exist in the form of what we call short tandem repeats, or STR for short. So STRs are basically a few nucleotides that keeps on repeating itself. So an example of that is CAG, 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 CAG. Okay, and the repeats can be as few times as 5 times or as many as 20 times, 40 times. It really depends on the location of these STRs, and the number of times it repeats is actually going to be different amongst different people within the population. And the difference is thought to be caused by the DNA polymerase. Actually, when the DNA polymerase tries to copy a region with short tandems, they sometimes made a mistake. It's easier to make mistakes in the STR regions because it's kind of like they lost track and they cannot keep track of how many times they've copied it already. So they might make a mistake and put a few extra CAG in when they copy it. So that's how the short tandem repeats might have arisen in the human populations. So sometimes the STRs are going to be a cause for concerns. It can actually cause certain diseases. And an example of that is the Huntington disease. Huntington disease is a neurodegenerative disorder that will ultimately degrade cognitive functions as well as a result in uncontrolled movements, and it has to do with the Huntington gene, okay, the HTT, and the mutation in the HTT involves the DNA segment with a lot of GAC in it. Okay, so this 3 nucleotides, we call it a trinucleotide repeat. So normally people would have the CAG repeated 10 to 35 times. However, in people with Huntington disease, the CAG segment is repeated 36 to more than 120 times. Okay, so this is an example of where the STR is going to be related to a disease. Okay, we don't want it to keep on increasing to very high number of repetitions. But not all STRs are related to diseases. Many STR sequences simply just exist within the genome itself, and we can use this STR polymorphism to identify a person. For example, if you have 2 DNA samples, how do you know it's from the same person or not, right? Let's say you recover a DNA sample at a crime scene and you have a few suspects. How do you know which one it belongs to? Well, you can sequence everybody's genome, okay, but that's going to be expensive, time-consuming, and it's going to be impractical. So instead of doing that, we can just analyze the STR sequences from these individuals, right? So first of all, we have to know where these STRs are within the genome, right? And people already know that because we have done the Human Genome Project, right? So we know exactly where these tandem repeats are going to be. So in the first person, we might have STR AGAT repeated 12 times, GATA repeated 15 times, and CCAC repeated 30 times. So this might be on chromosome 1, this might be on chromosome 5, and this might be on chromosome 13. So in another person, the same set of STRs will be found on the exact same location on the exact same chromosome. So on chromosome 1, chromosome 5, and chromosome 13. But this person is only going to be repeating AGAT 5 times, but repeat it GATA 15 times, CCAC 20 times. Okay, so even though they have the same number of repeats in one of the location, they are unlikely to have the same number of repeats in other locations. Okay, only people who are related or within the family would have very similar STR patterns, but for two completely unrelated individuals and for them to have the exact same STR in 3 separate locations, that's not going to be very likely. It could happen, but not likely. If we keep on analyzing it on multiple chromosomes, okay, maybe on 20 other chromosomes, for example, then it's virtually impossible for 2 people to have the exact same number of repeats in all those 20 locations. That's why the STR patterns are unique to every person, and we use the term DNA fingerprinting. So to analyze the STR sequence, we have to first understand something called gel electrophoresis. In gel electrophoresis, you are going to separate the DNA sample based on the size of a DNA segment. Okay, and what you do is you take some agarose powder and you dissolve it in a solution, and then you heat it up in a microwave, and after like a minute or so you take it out and you cast it onto a mold, and as it cools it's going to form a gel. And depending on how much agarose you use, the density of the gel is going to be different. So think of the gel as a network of fibers like this, okay, and the more dense it is, the harder it is for the DNA to kind of wiggle through it, okay. And if you use less agarose, then it's going to be more porous and DNA can move through it easier. The concentration of the agarose you want to use depends on the size of the DNA that you want to separate. So what happens is, after you make the gel, you're gonna put it in a buffer solution that contains various electrolytes in it, and that's going to help you conduct the current, because you're going to be hooking the apparatus to a power source. Okay, and over here on the top, this is where you're going to be loading your DNA sample. Okay, you can see there are many wells here. And you're loading your DNA sample. Okay, so DNA sample usually is mixed with a loading dye that kind of makes the sample heavier, so it will sink into the well, and you would use a pipette to put the sample in. And just to go back to this picture here, this is where you'll be loading the sample. Okay, so that's your DNA sample, and you will be running a current in this direction. Okay, in this direction, from the negative terminal to the positive terminal. You might remember that the DNA backbone, the sugar phosphate backbone, is negatively charged. That's why we want to run the current from the negative direction to the positive direction. So the current will actually be repelling the DNA, pushing them forward down the gel. Okay. So as time goes on, these dye along with the DNA sample is going to migrate down the gel as long as the current is running, and some is going to be migrating further than others. It all depends on the size of the DNA. Think about the gel again. Okay, I told you the gel is like a network of mishmash, and you're gonna have your DNA sample here. If your DNA is really long, then it's going to have a harder time kind of going through all these tiny holes, so they will be migrating slower. Whereas if you have a shorter DNA sequence, they will be able to go through this much quicker. So at the top here, right, the closer to the well, those would be the longer DNA. Okay, and at the bottom here, when you turn off the gel, when you stop the current, then the shorter DNA are going to be located farther down the lane. So after a while, it basically looks like this. The dye is not necessarily where the DNA is. Okay, the dye has the same molecular size, so they're all going to be going at the same pace, arriving at the same time, and you cannot actually see the DNA with your naked eye at this point. Okay, so in this well, you might have a long DNA here, and the next lane you might have a shorter DNA, and then over here you might have a really short DNA, and then another long one here. So there's no way to tell where the DNA is. Okay, the dye front, we call this dye front, That's just kind of give you a rough estimate of where your sample might be. Okay, if the whole diaphragm is all the way down here, then it means that you ran your gel for too long and your DNA sample is actually gone all the way out into the solution. So we don't want that. We want to cut the power when the diaphragm it's maybe in the halfway in the gel. So in order for us to visualize the DNA in the gel, we have to stain the gel. Okay, so we'll remove the gel from the buffer and we'll put it in a solution that contains something called ethidium bromide. So this is what ethidium bromide looks like. Ethidium bromide will bind to the DNA molecules. They actually will insert themselves between the nucleotides and they will fluoresce when you expose it to UV light. So because ethidium bromide inserts themselves into DNA, they are actually considered as a mutagen. Okay, you don't want to be handling this stuff without the proper protective equipment. So you want to wear gloves and you don't want to get it onto your clothing, so wear lab coats and stuff like that, right? But once you soak the gel in the ethidium bromide, you will be able to take the put the gel under a UV light, and you will be able to see exactly where the DNA are located. As a reference, usually people would load a ladder at the beginning and perhaps the end of the gel. And the ladder is something you can buy through commercial means, and it comes with DNA fragments that are predetermined size. Okay, so over here it might be like a 500 base pair, and then this one might be like a 450 base pair. Remember, as we go from the negative terminal to the positive terminal, the size of the DNA fragments are going to be smaller, right? So over here this might be like a 300, and over here this might be totally making this up, like 170 or something. So when you look at your sample and you see a very bright band here, then you know this is roughly about 172 base pair long. Okay, and you will know that this DNA sample here is maybe 268. Okay, so using the gel electrophoresis, you are able to determine the size of the DNA fragments that are in your sample. So now that we know how gel electrophoresis work, we will talk about how to combine that with short tandem repeats analysis to determine whether a sample, two samples are from the same person or not. So let's say we have three people here, and we have three separate chromosomes. Over here we have, let's say, chromosome 4, chromosome 7, and chromosome 18. Okay, so we'll be looking at the same three chromosomes for these three individuals. Okay, and on these chromosomes there would be some STR, some short tandem repeats. Let's say over here there is a short tandem repeats of whatever, let's say AT. Okay, AT, and for this person is repeated 20 times. Okay, so this is 20 times. And over here for this person, that same STR, the AT repeats on the same chromosome, is only going to be repeated, say, 5 times. And on this other person, it is going to be repeated also 5 times. Okay, and on chromosome 7 we are going to have another STR, another tandem repeats, and maybe it's over here this time. And for this person, let's give them some name. Okay, let's say that this is Bob. For Bob, this is going to be repeated, say 8 times, and for this person over here, we will call him Alex. Okay, it will be repeated 8 times, and over here for this third guy, we'll call him, I don't know, David or something, and that would be repeated, say, 15 times. Okay, on the last chromosome, for Bob, there is going to be a STR here, repeated 10 times. So this would be maybe like, CAG repeated 10 times. For Alex, it would be the same CAG, but it would be repeated, say, 20 times for him. And for David, it would be repeated, say, 5 times. Okay, so we would know the location of these tandems because, again, we have the Human Genome Project, right? So what you can do is you can take a sample from each of these people, and you can design PCR primers that would be flanking the tandem sequence. Okay, so for example, you know the sequence is going to be on the P arm of chromosome 4, and so you can design a primer over here and over here. So when you run the PCR, it will only amplify this section. Okay, and you would apply the same primer here for this person, for Alex, and you would be amplifying this section, and then you would do the same thing here. So you end up having a DNA clone of all the tandems. Okay, and you're going to do it for the same thing for chromosome 7 and chromosome 18. Of course, you're going to have different primers. Okay, you're going to have primers over here and here for chromosome 18, and you're going to have to design PCR primers for chromosome 8, chromosome 7 rather, on this end. Okay, so what you're going to have is you're going to end up having a mixture of clones of the red, blue, and green short tandem repeats. Once you've cloned the DNA, the next thing you want to do is you're going to want to run it on a gel. Okay, so this would be your agarose gel, and you would have some wells here. Okay, for simplicity, I'm just going to put 3 wells. Okay, you will probably lower leather here for reference, but again, we're just trying to focus on the samples we have. So if we load Bob's sample, okay, he's going to have sequences that are repeated 20 times, 8 times, and 10 times. So for him, he will have a band over here closer to the well that corresponds to this one. Okay, because it's very, very long, right? It's not going to be able to travel very far down the gel. For Alex, the same sequence, because it's much shorter than the one Bob has, it is going to be able to go much farther down in the gel. And for David, who happens to have the same STR repeats as Alex on this chromosome, it's going to travel to the same location. Okay, so if we were only to look at one single STR region, is not very informative. Okay, we can tell that okay, this person and this person are not the same person, but based on just one single location, it's hard to tell whether this is from the same person or not, right? They have the same tandem repeats, but you know, what about other location? That's why it's important to check other chromosomes as well. So in the same sample, right, we would mix all these DNA clones that we created in the same lane, and we load them, and for the this location for Bob, this one is repeated 8 times, so it's going to be able to go down quite far in the lane, right? And for Alex, it's repeated the same number of times, so you will be able to see the same band in the same location. And for David, it's repeated much more, right? Almost twice as many times as Bob and Alex, so that one is going to be higher up in the lane. Okay, the heavier DNA, the longer DNA, not heavier, the longer DNA is going to be closer to the well. And when you load the last sample, you will see that this is repeated 10 times, so it's going to be somewhere here perhaps, and this is 20 times, so it's going to be up here. And for David, is going to be repeated 5 times, so maybe it's going to be somewhere down here. Okay, so by looking at the location of the band, right, everybody is going to have a unique sequence, and so you will be able to compare the pattern to determine whether or not these people the samples are from the same person, and if the patterns are not the same, then you can say with high confidence that they're not from the same person. And in reality, we're not just going to be looking at 3 bands. Okay, we're going to be looking at you know, many hundreds and hundreds of bands, and so it creates kind of like a unique sequence that you can see and compare. Right, it's kind of like this picture that we have at the beginning of the video. Right, all these bands are basically the short tandem repeats that we're looking at, and by comparing it, we can say with high degree of confidence whether or not 2 samples are from the same person. In addition to STR analysis, you can also do something called RFLP analysis. So RFLP stands for Restriction Enzyme Length Polymorphism. So let's say you have a crime scene DNA and you have a suspect's DNA, and you want to see, if they're the same sample. So in RFLP, what you are going to do is you're going to mix the DNA with a restriction enzyme. Okay, so let's say this is the crime scene DNA, and there are two palindromic sequences that the restrictions enzymes are going to recognize. So if you mix this with the restrictions enzyme, it will cut it 2 times, creating 3 fragments. So when you run it on a gel, you will see 3 distinct bands. Okay, each corresponding to the DNA segment, right? So like 1, 2, 3 segments right there after you cut it. In the suspect's DNA, okay, because everybody genetic makeup is going to be different, and sometimes what happens is they have a single nucleotide substitution. Okay, so maybe this happens because of some kind of mutations that are a silent mutation, right? So it doesn't change anything, and it just caused the base pair to be different, and because of that difference in the base pair, when you expose this DNA sample to the same restriction enzyme, it will only be able to cut it once, right? You won't be able to cut this like the other one, because the palindromic sequence is no longer present. And so after treating this sample with the restriction enzyme and you lower it on a gel, you will only be able to see 2 bands. So based on this restriction enzyme length difference, you will be able to tell whether or not the 2 samples belongs to the same person. So we're just gonna end the lecture with gene therapy. The basic concept of gene therapy is to introduce genetic material into the patient's cells. There are 2 types of cells within the body. We have the somatic cells and we have the germ cells. Germ cells are sperm and eggs, and if you do germline gene therapy, you will be making modifications to the eggs and the sperm, which will affect all future generations from that patient. So this is highly controversial, right? Because you're making modifications on individuals that do not have a say about it, right? Because those individuals do not yet exist. Whereas if you make modifications in the somatic cells, then it will only affect that person, and the effects of the gene therapy will not be passed on to the patient's children. Typically speaking, the goal of gene therapy is to introduce genetic material that will allow you to either replace a non-functional gene, or you can suppress a gene that is overly active, or you can try to modify the clinical course of a condition. Let's say someone has a mutation in a gene that causes it to produce a protein that is no longer functional, right? Let's say they have a nonsense mutation and the protein is truncated, so it's not going to be able to work. So the logic here is to input a functional version of that gene, deliver it to the cell, and the cell is going to now be able to make the protein, and hopefully it will fix the condition. This is only successful if the effects of the disease are reversible, okay, or have not resulted in lasting damage to the body, right? So things like sickle cell anemia, once you give the proper copy of the hemoglobin gene, you should be able to start making functional hemoglobins, and the red cell should return to normal shape. Okay, there is a much more severe form of hemoglobin mutation called beta thalassemia. This is a nonsense mutation where a whole chunk of the hemoglobin protein is actually not synthesized. So people with this condition actually require constant blood transfusion to deal with the severe anemia, and because of the constant transfusion, they actually get iron overloaded as well. So in this case, the theory is you can replace it with a functional version of the gene, and the disease would go away. Okay, so same thing for hemophilia, right, which is a bleeding condition. If the disease already cause a severe problem, let's say someone has the lysosomal disease that we talked about, okay, and it already caused developmental problems, giving them a new gene is not going to reverse all those negative effects because it already happened, right? And you cannot make up for those delayed delayed development, so to speak. Okay, so it will only work for diseases that are reversible and have not resulted in lasting damage to the body. So the second application is to introduce a gene that would suppress an overly active gene. This is something common in cancer growth. Okay, an oncogene is a mutated gene that causes a protein to be turned on 100% of the time. So think back to the cell cycle. Right? Along the cell cycle, there are various checkpoints that governs how fast the cell will go through the cell cycle, and many of these checkpoints are proteins. So there are some proteins called growth promoter. Okay, so these growth promoters are going to encourage cells to go through the cell cycle. So under normal circumstances, the growth promoter will only be turned on if everything is normal. Okay, if the DNA are normal, if the cell is ready to divide, then the growth promoter will give the signal for the cell to progress through mitosis and divide. Okay, sometimes there are mutations in these growth promoters. So rather than only turning on when everything is okay, these things are turned on all the time, and as a result of that, it leads to rapid uncontrolled cell division. So in those cases, we would want to introduce a suppressing gene. So by inserting a gene into the cell with the faulty gene, this gene is going to be able to block the expression of the mutated gene. Okay, and so it resumes the normal cell activity. Finally, we can use gene therapy to modify the clinical course of a condition. The goal here is to insert a DNA into a diseased cell that will cause the cell to die. So maybe it's a cancer cell in a tumor, and by introducing these special suicide genes, so to speak, right, it's going to trigger cell death in the tumor, and that's going to allow the tumor to shrink. Perhaps in some cases, you can insert a gene that would create special markers to be expressed on the cell membrane of the cell, of the tumor cell, for example, and these markers can be designed in a way that the immune system would recognize and attack them. So again, this type of treatment would be ideal for cancer. Here is just a visual representation of that. We have the diseased cell here. By introducing the suicide gene, it would trigger self-destruction, apoptosis, in those diseased cells. And when the diseased cells all die, presumably the condition of the disease would improve. In another example, we would have the diseased cells, and we would introduce our marker gene, and the marker gene would cause the cell to express certain types of marker proteins that will be recognized by your immune system, and they will attack and destroy those cells. There are two ways you can do gene therapy. In ex vivo, you are going to be extracting the cells from the patient's body, such as the bone marrow cell in the case of leukemia gene therapy, for example, and you will be making modifications to those cells in a petri dish in a laboratory, and then once the cell grows a little bit, you will reimplant them back into the patient. On the other hand, you can do it in vivo. In vivo means directly in the person's body, and this time you're going to be using a vector, something that will carry the DNA, the gene that you want to insert, and the vector will help you distribute that gene to their target. There are several challenges in gene therapy: target delivery, immune response, interaction with other genes, as well as cost. Target delivery. It's very important that we deliver the gene to the exact right cell and in the exact right place. If we deliver the gene to the wrong cell, that would be inefficient for one, but it could also cause other health problems. Let's say we wanted to deliver the gene to the cancer cells so that they express certain proteins for the immune system to attack. If that ends up in your healthy cells, then you would have a problem, because your immune cells is going to attack your healthy cells now, right? So the target needs to be very precise, and we need to have a delivery method that would be on target. even the when the gene gets to the right cell, they have to be able to act on the right gene. Okay, you have to be able to turn on the gene, and sometimes in order to turn on the gene, you have to insert the gene, and we need it to be inserted into the right place. Okay, we don't want it to insert into the middle of another gene, right? And that's going to be problematic. And in fact, sometimes the cells they have built-in mechanisms to shut down foreign genes that are showing unusual activities. So even though the gene may get into the cell, there will be other mechanisms that will shut it off. Okay, so there are all these problems that still needs to be solved for gene therapy to be developed widely. Here is an example, right? We can use a virus as a vector. So we can take something like a cold virus, and we take away all the parts that would cause you to become sick, and we would give the gene to the virus, and then you would infect the person with the virus, and the virus would take the gene into the cytoplasm of the cell and deliver it into the nucleus. So that at least is in theory how it should go. And sometimes because you are using a virus, then there is going to be some kind of immune response. Okay, so the new gene introduced by the gene therapy could potentially be considered as harmful intruders to the immune system, and the immune system would respond accordingly, and it could respond in a way that could harm the patient. So the challenge is to find a way to deliver the genes without triggering the immune system. Even if everything went well, the gene was delivered, there was no immune attack to the delivery method, the gene was not suppressed by the cell zone defense machinery, there would still be the concern of interaction with other genes. There is a risk that the new gene will insert itself into the path of another gene and therefore disrupting its activity. And when that happens, it could lead to damaging effects. For example, if you're trying to insert a gene to replace a loss of function of a gene, and you end up interfering with another gene that regulates cell division, that could end up resulting in cancer. And the final challenge is the cost. Many of these genetic disorders that could potentially be cured by gene therapies are extremely rare. That means not a lot of people in the world have these diseases. So the gene therapy for those diseases, for those people, are going to have to be very individualized and tailored. And because of this, the cost associated with it is extremely costly, and it could cost as much as $1 million per treatment. But there are many examples of successful gene therapy that has been performed to date, and it's still a very new field of research, and there needs to be a lot more work to be done. But I believe something like this is going to mature over the next several decades, and eventually this might become the norm of how diseases are going to be treated. And recently there are the discovery of a new technique called CRISPR gene editing tool, which basically allows you to make any changes in the DNA sequence at a very specific location. We don't have time to go into the details for the CRISPR technology, but I'll post a link online for you to look at that if you're interested. And with some powerful tools like that, it would make the design and the changes in the genetic sequence much more easier, and potentially making gene therapy much more effective and accessible. That's it for the lecture on biotechnologies. Thank you for watching.